The arrangement of membrane proteins. The main basic thing is they're integral and peripheral proteins. The integral ones, they're going in, sometimes even completely through, when they are transmembrane proteins, and when they're the peripheral proteins, they're just on the outside. And some which are spanned through, they are having then an alpha helix, and if it goes many times back and forth, it has then a hydrophilic polar region which is protruding into the fluid, extracellular fluid, the watery part, and the watery part, the cytosol, inside of a cell of a lipid bilayer. And also they are empathetic, so one part of them is hydrophilic, and the other one is hydrophobic. The hydrophobic part is in the center, if they're transmembrane, for example, and protruding in them. And that adds to the asymmetry of the lipid bilayer, that the outside is different from the inside part. Also, the transmembrane proteins also are differently on the inside than on the outside. And all these glycolipids, which are the lipid bilayer, the glycolipids, with their head and their fatty acid tail, which are non-polar in the center and hydrophobic, they are now these glycoproteins also, and they have also a carbon group attached, 2 to 60 monosaccharides, and they add to that whole thing of having the outside a sugary coat of carbohydrates, of these monosaccharides. And that forms a completely a sugary coat, the glycocalyx. And that helps to detect which cells are foreign and have to be destroyed for the human body and which are not. It helps to make them slippery because they are hydrophilic. They at the outside, they're hydrophilic, they are pulling in water. And so it prevents them from drying out in the airways in the gastrointestinal tract and makes them slippery if it are blood cells. And that is according to Gerard Roggan-Senner-Grabowski's Principles of Anatomy and Physiology book on the page 63, but this is no substitute for medical knowledge and also no medical advice. I'm Rebecca. I'm trying to learn cytology today, the arrangement of membrane proteins, and after we've done it, we're going to do Armstrength training. And if you participate in the Armstrength training, then it's already Armstrength. Arrangement of membrane proteins. Membrane proteins are divided into two categories, integral and peripheral, according to whether or not they are embedded in the membrane. So there are some proteins, some are integral, others peripheral, whether they are part, inside, or whatever of the membrane. Integral proteins extend into or through the lipid bilayer among the fatty acid tails, and can be removed only by methods that disrupt membrane structures. Integral going into or even through the lipid bilayer. Peripheral proteins, on the other hand, associated with membrane lipids or integral proteins at the inner or outer surface of the membrane and can be stripped away from the membrane by methods that do not disrupt membrane integrity. Okay, the integral, they're in or completely through and can't be removed without disrupting the membrane structure. Peripheral, they are just at. I think peripheral, the word something means like out of the periphery. It means something like that. They are at the surface, could be at both sides, of a lipid duo bilayer membrane, and they can be removed or going away without disrupting the integrity of the membrane. Like membrane lipids, integral membrane proteins are empathetic. Okay, one part loves water, the other one not. They are hydrophilic, that one loves water. The regions protrude into either the aqueous extracellular fluid or the cytosol, and the hydrophobic regions extend among the fatty acid tails. The integral proteins. The integral proteins, one part of them loves water and it protrudes into the extracellular watery fluid at the outside of the cell, or the cytosol, the fluid part of the cell inside, and the hydrophobic part which doesn't want water of Integral proteins is in the center in the hydrophobic region, the non-polar hydrophobic region of the lipid bilayer. Integral proteins that stretch across the entire bilayer and project on both sides of the membrane are termed transmembrane proteins. So that's all. Some proteins are going from outside the cell through the first layer, through the center of the layer, through the second layer, and protruding into the other side inside the cell. They are transmembrane proteins. Often the portion of protein that spans the nonpolar portion of the membrane consists of an alpha helix that is formed by amino acids having mainly nonpolar hydrophobic side chains. I don't really know what I want to tell us with that, but I now understand what they are telling us. And that is, if that thing is spanning it, in the center that's spanning through, it is a nonpolar, like the lipid bilayer is in the center nonpolar, and also hydrophobic. It's hydrophobic, there's this dry in the center, the water is in and out. Hydrophobic nonpolar amino acids, which are alpha helix, consisting out of alpha helixes, alpha helixes, which are amino acids which are nonpolar hydrophobic. In some cases, the protein's amino acid chain loops back and forth across the membrane several times. And there can be such a hydrophobic. How is it working? It's when even the part which is not in it hydrophobic, which is not in the center, also hydrophobic and nonpolar. And it can be that of such an integral transmembrane protein, the alpha helix consisting out of amino acids is going multiple times from side to side. Hydrophobic alpha helix segments that span the membrane alternate with polar hydrophilic portions that protrude into the aqueous fluid on either side. There we have it. There are polar and hydrophilic portions of the alpha helix amino acid thing, which are where it protrudes into the aqueous fluid on the other side. And here I answered the question. If it goes back and forth, this alpha helix consisting out of amino acids, they are then at the portion where we're going through it, polar and hydrophilic. Although many of the integral proteins can float laterally in the lipid bilayer, each individual protein has a specific orientation with respect to the inside and outside faces of the membrane, thus contributing to the membrane's asymmetry. I don't really understand what we mean with lateral, but what we mean is also if I spin completely through it, the side which is on the inside to the cell or to the outside is different, and therewith they are also one of these things which make the lipid bilayer asymmetric. It's not the same to the in and outside. Many integral proteins are glucoproteins. Proteins with sugary carbohydrate groups, which are glycolipids. Now we have glucoproteins. With sugary carbohydrate groups attached to the ends that protrude into the extracellular fluid. So just like the glucolipids, which have a sugary carbohydrate group attached, which is hydrophilic and polar and protrudes into the water more or less, the glucoproteins also have a sugar carbohydrate group attached at one end, which is protruding into the extracellular fluid, and is probably also hydrophilic and polar. These carbohydrates, which are attached, are short, straight or branched chains composed of 2 to 60 monosaccharides. Glucoprotein. And these carbohydrates can have short, straight or branched chains. They can be 2 to 60 monosaccharides. And the monosaccharide we did within biochemistry. These are 3 to 7 carbons, each of them with a water molecule basically. And they have in glucose or fructose, for example. The carbohydrate portion of glycolipids and glycoproteins form an extensive sugary coat called the glucose, which has several important functions. The glycolipids and the glycoproteins both have at the end a sugary carbohydrate group, and they are hydrophilic and polar, and that gives without must say what, it an extensive sugary coat, and that has been called combined glucocalyx. The composition of a glucocalyx acts like a monocular signature that enables cells to recognize one another. For example, a white blood cell's ability to detect a foreign glucocalyx is one basis of immune response that helps the body to destroy invading organisms. And the way they are structured, composed, ordered, sorted, these glucocalyx out of the sugar heads of glucoproteins and glycolipids helps the white blood cells to detect which cell is one of their own body or a foreign one, and helps to tell the human body which one to destroy. In addition, the glucocalyx enables cells to adhere to one another in some tissue. I don't know what adhere means. I think adding to it. And it protects the cell from being digested by enzymes in the extracellular fluid. Sometimes they can adhere to them, I think that means bring them together, adhere to another one, and also are protected from being digested through the glucocalyx, that sugary part of the glycolipids and glucoproteins. The hydrophilic properties of the glucocalyx attract a film of fluid to the surface of many cells, an action that both makes red blood cells slippery, as they flow through narrow blood vessels, and protect cells that line the airways and the gastrointestinal tract from drying out. So this hydrophilic portion of this glucocalyx is attracting water because it's hydrophilic, and that makes the red blood cells inside the blood slippery so that they can go through the blood vessels, and also cells in the airways and inside the gastrointestinal tract are not drying out because they are attracting a fluid or watery thing. That was the cytology part. If you were here for cytology, you can go directly to the next part of cytology, where we try to make a little presentation about the arrangement of membrane proteins. If you stay here and participate in the Armstrength training, then it's really honest. Good and stable stance, and breathe in and reach with the hands up. Reach with the fingertips as far to the sides as possible, and again down, reach again. And every time you reach, and down. Reach in front. Back, get in front. And no. Long arm, strong fist. If this exercise causes pain or discomfort, then don't do it. Long arm, strong fist, and circle the fist. And change the direction. Now to the next exercise. Press the fingertips together. Have a little bend in the elbows. And you don't have to do it with full force. Your arms are stronger than your fingers can hold. And if there's pain or discomfort at the elbows, wrists, fingers, or in particular the finger joints, and stop immediately. And next exercise, now with the fingertips. And again, if there's at any time pain, then stop immediately. And you don't have to do it with full force. And next exercise, press with the fingers into the thenar and hypophynar of the hand. If this causes damage at the skin or at the fingernails, not good. Press from the bottom and give it. Fine. Press and give it. If this causes discomfort at the wrist or something, if the angle doesn't work when done, press. Change. Press. Give in. And press. And now the other hand from the top. And give in. And press. Now it's round. And one last of this time, the hand out. Yes. Yes. Now press with the hands together and go over to one side. Up. Full force isn't necessary. And if anything of that causes discomfort or pain, then don't do it. Over again. We don't have much time left, so don't wait. Over, up, over, down, change again. And relax the hands, circle the arms in front. Backwards, one arm in front. Relax the arm and pull it in. The other arm. And go with moderate force over the middle part of the lower arm. And that was the arm strength training. I thank you very much, and until next time.